Alright, so the food tests you need to know are, you need to know a test for a protein. And this is any protein. So what you're going to add is a reagent called Biuret reagent. And by Biuret reagent, you're going to add it by, here is your sample, you're going to drip it down the sides of the test tube, and as it goes into here, it will give you a lilac or mauve colour. Okay? So the positive test is the colour change from blue to a lilac or mauve colour. It's not it goes lilac, it's that there is a colour change from blue, which is the colour of the Biuret reagent, to a lilac or mauve colour. If it's a negative result, it is not that there is no change, it's that it remains a blue colour. Yeah? So the reagent remains a blue colour. It's very clear that whenever you're describing the change, you describe the change, not just say it didn't work or something. Right, the next one is a test for starch. And starch, remember, is a polysaccharide you find in plants. And the way you test for starch is you add iodine in potassium iodide. And you take your solution, you put a few drips in it. Now iodine in potassium iodide is a yellow colour. You drip it in, and if starch is present, this thing will go a blue-black. So the presence of starch is the colour change from yellow to a blue-black colour. And if there's no starch present, it remains yellow colour. Okay, the test for a fat is the emulsion test. And in the emulsion test, you take fat, you shake it with alcohol, you shake it with say 5 centimetre cubed of alcohol for 1 minute. And alcohol is a non-polar solvent, so it will be dissolving the fat that's present. You then pour it into water, and as you pour into water, it should make a creamy white emulsion. If it makes a creamy white emulsion, then it tells you that a lipid or a fat is present. Next test is the reducing sugars test, which is for monosaccharides like glucose and fructose. Now, this is with the Benedicts reagent. Now, Benedicts test involves taking copper 2. So Benedicts test contains copper 2, which is blue. When you boil, so it's definitely boil, not heat. So when you boil the sample you're testing with Benedicts, and you're going to boil it for 5 minutes, then the copper 2 will be reduced to copper 1 if a reducing sugar is present. And the copper 1 is a reddy browny colour. Now, the tests we've looked at before have been qualitative tests. They've been tests for the presence or absence of things. So, starch is a qualitative test, the fat test is qualitative, the protein test is qualitative. Now, Benedicts is a semi-quantitative test because depending on how much of the Benedicts reagent is present, depends on how red it goes. So if you only, sorry, not how much of the Benedicts reagent, sorry, how much of the reducing sugar is present. So if you have a small amount of a reducing sugar present, then it will produce a few reds. So it will be a few reds and blue, which will give you a greeny colour. If you have a bit more glucose present, or any other reducing sugar, then it will be like a browny colour. If you have an even more present, it will be a brick red, dark red colour. So this is what makes it a semi-quantitative test, because the degree to which the colour changes tells you how much is present. Now, the final test is the non-reducing sugars test. So this is for non-reducing sugars, such as sucrose, which is the sugar you put in your tea. Now, sucrose is a disaccharide, and it's a disaccharide of glucose with a glycosidic bond to a fructose. Now, a non-reducing sugar is one that when you boil it with Benedicts, it does not change colour. So you take a fresh sample and you boil it with hydrochloric acid. Now, boiling it with hydrochloric acid will break this glycosidic bond. When you break the glycosidic bond, you're then turning these two into reducing sugars. So you then have to neutralise the hydrochloric acid, and you do that by adding sodium hydrogen carbonate and testing with a piece of universal indicator paper to make sure it has been neutralised. And then you boil with Benedicts reagent, which means that it will now test positive because these two are now reducing sugars. So the non-reducing sugar test is a sugar that tests negative with Benedicts, but you then take a fresh sample, boil it with acid, neutralise it with sodium hydrogen carbonate, then boil it with Benedicts, and then it tests positive. Because by boiling it with acid, you're turning these non-reducing sugars into a reducing sugar, which then changes the Benedicts and turns it into the red, which means you can then detect it.